Well, hey there, here's a very quick mini tutorial using some sketch notes that I just created on carbohydrates. So understand that carbohydrates are macromolecules. You know that already, so this is just a class of macromolecules, sometimes called biomolecules because it has to do with biology. And they have a lot of important functions. And I have three of them here, so providing energy, also storing energy, helping to build needed compounds in the body, so they serve as raw materials for that. And they provide certain structures, structural components that we're going to see. So carbohydrates, you can see here, we can discuss them a lot of different ways. The word itself, carbo, has to do with carbon, hydrate has to do with water. So the formula here, you can see the condensed version of it is a 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. So that's kind of handy. Then you can also see that we can classify them according to the number of units they have. So if monomers are single units and polymers are bigger units, multiple units, monosaccharides are the units for carbohydrates, because saccharide is referring to these things we know as sugars. Polysaccharides are the polymer version of sugars, right? So we have polymers, many units. Polysaccharides are the things that we typically look at as starches. So we'll look at that in a sec. So, but to form these larger units from the monomer units, that happens through dehydration synthesis. So we're going to lose water every time we form a new bond. And we're also going to add energy in, which I don't have here, for the formation of that bond. So lots of ways to classify, and I actually don't have every detail on here because I want you to think about it as we go. But one of the ways we can classify is just simply by where is that functional group for carbohydrates. So the main one for carbohydrates that we see is this carbonyl group. And the carbonyl group can be either on the very top of a chain, if you want to see it as a chain structure, or it can be within the structure. And if it's at the top of the chain, think about what you would call that. What type of carbohydrate or formation is that? Think for a second. Okay, would be the aldehyde version, therefore this would be an aldose, OSE type of sugar. If it's down here, it's a little further down, we call that the ketose type, and therefore our ketone carbonyl arrangement here, but it's the ketose when we say it's a sugar. And there you go. Now these also form rings, so we can see them in a linear form, we can also see them in a ring form. So I just wanted to show those to you, with A corresponding to this one right here, and B, and here it is in the ring form, and B over here. But I also counted the carbons here, so you can see the carbon numbers. If it's in that ring structure here in from the aldose sugar, we count this carbon right here where you see the colorful yellow as that carbon number 1. And that kind of tells us where this bond is forming. Literally it's forming here between this first carbon here, and when it kind of curls around to join, it's joining with this 5th carbon on the chain, and then that 6th carbon you can see is sticking up above the plane of the ring. Where over here in this other version, this has got a different shape, right, a pentagon shape. This is actually fructose, and you can see that number 1 carbon isn't involved with that sort of ring. It's sort of sticking up again above the plane of the ring. It's that 2nd carbon, and notice where that is over here, the 2nd carbon. Here's the double bonded oxygen, the carbonyl group there. So that's just a different way to look at it. And normally we find a lot of carbohydrates cyclize in the body. So they'll go from a chain linear structure to a cyclic version or ring structure. So think about also some other versions here of ways of classifying. So by the number of carbons. Now I didn't put anything here, but you could come up with that. So if it has 3 carbons, it would be a triose. If it has 4 carbons, we don't see that as often, but we certainly see 5 and 6 carbon structures. We can also look at it in terms of the number of joined individual units, right? So we can see things which are on their own, just the single monomer structure here would be a monosaccharide. If there are two of them together, disaccharide. When we get more complicated and we get bunches of them together, which are polysaccharides. And then we can get a little more specific on some of these. I did not get specific here, but you should know a couple of different monosaccharides. You should know a couple of disaccharides. And here on the polysaccharides, because I want to make some distinctions here, I actually tell you a little bit about them. So those which are joining together through these alpha glycosidic linkages, which we've talked about, and form these straight chains of that. So they're going to form these alpha 1 to 4 glycosidic linkages. When those happen, and they have straight chain, that kind of describes this one we see here, amylose. They can also branch, though, and this is the version that we talked about with amylopectin, where you have some of it straight with those 1 to 4 linkages, and some has some type of branch point, which is a 1 to 6 linkage to form the branch. One that wasn't a picture in the book, but is actually even really much more important in a lot of ways, is the glycogen, which also does the same type of thing that amylopectin does, but it's more branched. It's much more highly branched. And why glycogen is important, I put a little star down here, is because it's a storage form of energy that's stored in muscle tissue. But all of these are energy sources and can be used to increase blood sugar levels, and we'll see that later on. Now, there are also these things called beta linkages, and we talked about that, and here's one. I tried to show it in sort of blob form. So beta linkages are going to be formed in a reverse type of linkage than we normally see, and this example here in particular is cellulose, and cellulose is a type of polysaccharide that forms this way, and the fact that we have no enzymes for these types of beta glycosidic linkages just means that we can't break them down. We can't extract energy from these bonds. But they are really important because they form it's the main structure of plant cell walls. And that's it. Hopefully that made some sense.